Hello and welcome to another lecture. Today we are talking about DNA technology. So let's take a look at what we discussed last time. Last time we talked about DNA synthesis, and you will see that today will also be about DNA synthesis, except today we're talking about synthesis in the lab, in the test tube, whereas last time we talked about DNA synthesis in the cell. So we said that DNA is synthesized in a semi-conservative manner. We described what that is. We looked at the original experiments that led to the discovery that, in fact, it was a semi-conservative process. We said that the growing DNA molecule grows from its 5' to its 3' end, because that is the only way that the polymerases can add the new nucleotides. And that was similar in the RNA molecules as well. We said that due to the anti-parallel nature of the DNA, the two strands are synthesized in opposite direction. Right? Anti-parallel means 5', 3' on one end and 3', 5' on the other end, or inverse to each other. The two strands therefore differ. One can grow continuously, and the other one has to be made in sections, and those sections are called the Okazaki fragments. Synthesis requires RNA primers, the primase and the primers, the enzyme versus the piece of nucleotides, topoisomerase enzyme, the helicase, the polymerase, ligase, and exonuclease, all enzymes ending in A's. DNA polymerase has DNA proofreading capability, and telomerase helps with the finishing of the synthesis at the end of the linear chromosomes. So now today, let's take a look at how DNA synthesis is done in the lab and why we want to do it and how we can do it. So I have talked about this a few times already, and I've always said we'll discuss this at a future lecture. Well, now is the time when we'll discuss this in this lecture. And so this process is called polymerase chain reaction, or short PCR. And that is a technique to amplify small sections of DNA. So it's not the entire genome that gets amplified. From single or just a few copies to a large amount. So if you just have a speck of a sample, a little bit of DNA extract, or a bit of saliva, or a blood sample, or even just the root of a hair, you can take that amount of DNA and you can amplify it so that you can then analyze it. That very small amount of DNA is usually not enough to do extensive analysis with. So that's why the development of the PCR was so monumental, because you could now do so many more things with DNA that you couldn't do before. So let's see how DNA synthesis occurs in the test tube in this process. So the process is broken down into 3 steps. The steps are called denaturation, annealing, and extension. And this goes in multiple rounds, about 30, sometimes 40 rounds of denaturation, annealing, extension. And then you go back to denaturation, annealing, and extension. And in every cycle, every time that you cycle around in these 3 steps, more and more and more DNA is made. So in the DNA part, denaturation part of the amplification, the solution containing the double-stranded DNA is heated, and the DNA strands are melted apart or denatured. So the 2 strands are broken apart. The hydrogen bonds that hold them together are overcome by actual physical heat. So there's no enzyme that's used here. This is done by brute force in the lab. So now you have your 2 strands of your target. And let's say that target is a particular gene. It's just maybe a few hundred, maybe 1,000 base pairs long. So it's not much longer than that, what normally gets amplified in PCR. So now you have your pieces, and you bracket that piece of DNA that you want to amplify, let's say that gene that you're interested in, by adding small pieces of primers. So those are, again, little pieces of DNA that are complementary to a stretch of DNA on the target, similar to the primers that were needed in order to start the amplification or the synthesis the synthesis procedure during DNA synthesis. Remember that also required an RNA primer and primase. Well, this requires a DNA primer. It's about the same size, same idea. You have a piece of nucleotides that is complementary to a piece of DNA that you then want to amplify. So this piece will bind to this strand, and that piece will bind to the other strand. Each of them have to be complementary to the piece that they will bind to. So that binding is also referred to as annealing. Annealing means to bind. So that is the annealing phase. And then you have the extension phase. Now you add a special kind of DNA polymerase, one that can live at these relatively high temperatures. We'll come back to what exactly those temperatures are in a second. But it's a DNA polymerase that makes DNA in an organism that usually lives in hot springs or very warm areas, and so it can withstand this heat. And at that point, it then amplifies the DNA and just amplifies everything that's in its way from here on out. It eventually will fall off, but it will shoot past the stretch that you actually want to amplify initially. You will see that in the next round, when the purple piece becomes the template, now that has a defined end. And so therefore, when you, in the second round, I'll show you that in a second. In the second round, when you use this piece as the template, you go in this direction. So then you amplify this piece and it will fall off at the defined end of the bracketed piece of DNA that you want to amplify. All right, we'll come back to that in a second. So this slide, I want you to see that we need 3 steps: denaturing, annealing, and extending. So let's see how that works in the step-by-step process with all these cycles. So if you have your target sequence, 5' to 3', 3' to 5', then you get your primers, you denature the DNA. That gets usually done at 96 degrees Celsius or 94 degrees Celsius, so almost boiling. The primers are annealed, so they're added on, and then in each round of amplification, you double the number of molecules that you have. So here you start with your target, you're making 1 copy, and now you have 2 copies. And so then when you go back to the first part where you now denature this part, and then you anneal in the next round, the second round of annealing, there's more primers, and they can now anneal, and they make a copy, and then you extend, and then the third round starts, and you get more and more and more copies every time. It's much easier to see that in an animation. So there's so many animations online in which you can see this, and I would recommend to you to just go online, look on YouTube or other places where you can see multiple PCR animations. I found one here that's short, and it comes from a site that has lots of interesting stuff. It's called the Cold Spring Harbor Learning Center, DNA Learning Center. And let me just play this for you.
Polymerase chain reaction, or PCR, uses repeated cycles of heating and cooling to make many copies of a specific region of DNA. First, the temperature is raised to near boiling, causing the double-stranded DNA to separate or denature into single strands. When the temperature is decreased, short DNA sequences known as primers bind or anneal to complementary matches on the target DNA sequence. The primers bracket the target sequence to be copied. At a slightly higher temperature, the enzyme Taq polymerase, shown here in blue, binds to the primed sequences and adds nucleotides to extend the second strand.
So just pausing it here for a second, she said Taq polymerase. That is an abbreviation for the name of the organism from which this DNA polymerase was harvested originally. So it's a DNA polymerase, but it has the special name Taq polymerase.
This completes the first cycle.
Okay, let me pause it again. So see how the first cycle still has the long ends, and then it has the short ends where the original PCR primer bound over here, make this copy, but then overshot, right? This was the goal to be stopping it, but it overshot. So now we have 2 pieces here, 2 new pieces. They are longer than the bracketed stretch of DNA that we originally want. We have the original pieces, and now we have the first round pieces, and they are a little bit too long. see what happens in the next cycle.
In subsequent cycles, the process of denaturing, annealing, and extending are repeated to make additional DNA copies.
All right, so now you see this piece is the original, is the length of the bracketed piece, the targeted piece of DNA that you want. So now in the third cycle, you're starting to get pieces that have a very defined start and end point. We still have the original templates, and we still have the pieces that are a little too long from the first round, but you will see that after a few more cycles, those few copies will not even be noticeable, because there will be millions, literally millions of correct size pieces.
After 3 cycles, the target sequence defined by the primers begins to accumulate. After 30 cycles, as many as 1 billion copies of the target sequence are produced from a single starting molecule.
Okay.
Polymerase chain.
Okay, so here we have it. So that is DNA polymerase chain reaction. Now, you have these pieces and they're in a tube, and you will actually do this in lab. So you will see that you have a little plastic tube with very small amount of liquid that's clear, and that now supposedly has all your DNA that's amplified. And the question is, how do you see that? How do you see that it's a piece of a given size, a certain stretch of DNA? How do you know that you have it and nothing else, or how many more pieces you have in there, whether you have anything or nothing? That all gets done in a technique that's called gel electrophoresis. And we talked about that very briefly previously. I want to bring it back here for a second. So gel electrophoresis uses electricity here and here. So you have the positive pole here, the negative pole over here. You have a box, and you have a tray on which you have a gel slab. And so imagine that this is a piece of Jell-O, and imagine that the piece of Jell-O was poured by, taking the Jell-O out of the box, adding hot water, and then stirring it and dissolving the Jell-O. And then when it solidifies, it makes this Jell-O-y substance. Well, if you pour it into a dish, then that will give it a shape, right? And so that's the same idea with a tray. The tray will give this Jell-O slab this square gel shape. And then I said, in order to make little dents into the gel, if you imagine that you left the spoon in there, and then after solidification you take out the spoon, you have a hole in the gel. That's what gets done in a gel electrophoresis agarose gel as well. So they don't use gelatin, they use a substance that's called agarose. So in agarose gel, you put in these little combs, so basically little pieces that will leave a hole when you take it out after solidification, and that's where you then load the DNA. So these are called sample wells. You add the sample, and then this is one lane. This is called a lane of DNA. So you can, on this gel, run, 8 lanes with 8 different samples. And then along the lane, you see the separation of DNA pieces of different sizes, with the largest size at the top of the gel that has taken the most time to start wiggling itself through the gel. And the smallest piece is at the end of the gel. That runs fastest through the gel towards the positive pole of the positive electrode here, because the DNA is negatively charged. So that is what's pulling the DNA through the gel. There's a liquid in here, a buffer, that will transmit the electricity through the gel to the DNA pieces. And so this piece here, this setup, is what you see amplified over here. Well, not really amplified. You see it in a real photograph. So you see a photograph of this gel after it was separated. And you can see that each of the pieces are visible because there's a light, a fluorescent light that is shined on top of them. And the DNA is not visible by itself. So you have to add a dye that binds to the DNA, and only when it binds will it fluoresce when excited with UV light. So you shine UV light on it, the dye that is intercalated into the DNA will then fluoresce and will tell you where in the gel you have pieces of DNA. And so now you can see large pieces of DNA over here, small pieces of DNA over here. This is not exactly what a PCR gel will look like. A PCR gel usually only has one piece, one very specific size of DNA, because you design the primers such that you know how long that targeted piece is going to be. So let's say maybe 300 base pairs. You would only then have a gel in which you have a 300 base pair band here. And maybe if you ran a similar PCR reaction with the same primers, you would have another 300 base pair band over here in the next sample. So this would be an example of multiple pieces of DNA that were all run in the same lane. Okay, so over here on the right-hand side, I have just summarized what I just told you. Single molecules cannot be seen by gel electrophoresis. You have to make millions of pieces by PCR so that you can see it. The DNA is then stained with fluorescent dye. The dye intercalates into the DNA. The larger pieces are higher up, the smaller pieces are further down. And then I say here, what you can do in order to determine what the size of a stretch of DNA is, these are called bands on a lane of DNA. So if you want to know what the size of this band is, the only way how you could do that is by having a reference of some sort. And so you can load at the same time that you load your unknown piece of DNA into this well. Next to it, you could load known pieces of DNA. And those are called DNA ladders. They're called ladders because this almost looks like a ladder, sort of rungs on a ladder. If you know that you put in a sample that has pieces that are, let's say, 300 base pairs, 500, 700, 1 kb, 1,000 base pairs, 2,000 base pairs, 3,000 base pairs, and so forth, then you know what each piece is, if you know that. And then you can say, okay, if this is 300 base pairs, I know that because it says that on the package where you buy the DNA ladder, which is a preparation of DNA of known sizes, then you can say, all right, so this is 300 and this is 1,000, then this has to be maybe 500 and 600 or something like that, right? You can, from the known pieces, estimate the unknown pieces. Okay, so that is gel electrophoresis, and you will do that, you will use that in lab as well to separate the pieces of DNA that you will get from your own PCR reactions. So a very important tool for a biotechnology lab is the use of restriction enzymes. We've also already mentioned restriction enzymes before in this class. Restriction enzymes are enzymes that cut DNA. They restrict them, the pieces of DNA. That's why they're called restriction enzymes. That basically means it cuts it, but it doesn't cut it randomly and furiously everywhere. It cuts it once at a very particular sequence. And each type of restriction enzyme, they all have their own names. The names are made up by using the first name of the first letter of the name of the bacterium from which they were originally isolated. So there's one that's called EcoR1, and that stands for E. coli or Escherichia coli. That's a gut bacterium. And that bacterium has this particular enzyme, and they use that enzyme to cut invading DNA from viruses and other bacteria when other bacteria try to invade them. And so they have these enzymes in order to cut up foreign DNA. So that means they don't have that sequence themselves. They have an enzyme that will cut a specific sequence in the invading DNA. And so there are all these different kinds of bacteria out there, and people have now sampled so many different types of restriction enzymes that you can almost find one particular restriction enzyme for any kind of DNA sequence context that you can imagine. So what do I mean with that? Here's a sequence context, GGATCC. So if you find that sequence and you want to cut it here where the arrows are, between the G and between the G here, then you can use an enzyme that's called BAMH1. And in order to get BAMH1, you go to a online catalog for a company, biotechnology company that sells restriction enzymes, and you say, well, let's see, do they have BAM-H1? Yes, they do. You buy a little tube of it, and now you can, with that enzyme, cut this particular sequence. And that's the same with all these other enzymes, KPN1, HPA1, Hindi3. Some of them have funny names, SMA1, and so forth. And you can see that some of the enzymes produce what's called a blunt end, blunt because on both ends it's the same length, and some of them produce what's called an overhang. So this is an overhang because the lower part of the DNA gets cut here between the C and the T, and the upper part gets cut between the T and the C here. So that means this piece hangs over and this piece hangs over because, of course, the DNA goes on here, right? It's double-stranded over here, and now you have the top strand hanging over a little bit over the bottom strand as these two pieces separate from each other. And the bottom strand from this part over here on the right-hand side, that hangs over a little bit from the other side. So those are called sticky ends because they have a T or a C or a G in a nucleotide that would like to bind with another nucleotide through hydrogen bonds. Blunt-ended enzymes are not sticky. All right, so restriction enzymes are enzymes that are originally isolated from bacteria where they serve as a way to defend themselves, defense mechanism. But in biotechnology, they are used to cut DNA at very specific sites. So I wrote down here on the side that you can use these restriction enzymes, and that's the context in which we have discussed them in class, to create either restriction enzyme length polymorphisms, RFLPs. If you look back 2 lectures, we talked about RFLPs. And I also say here that if you have a SNP, a single nucleotide polymorphism, so that means difference between the 2 different copies of a piece of DNA, for example, a gene, you can use a restriction enzyme to create SNP markers, to use the different allele sequences to cut either DNA from one allele and not cut it from the other, or vice versa. And so then you can use that restriction enzyme to create a restriction fragment polymorphism. So if you look back to, I think it was 2 lectures ago, we talked about making these markers, using these markers, what a SNP is, what an RFLP is. In that context, I talked about restriction enzymes, and so here we have them again. Restriction enzymes can also create identical cut sites on the ends of 2 DNA molecules. And so when you take this piece, right, and you cut it so that the left end and the right end are separated, then you can imagine that you could put another piece in between those now opening this. Let's say you have a piece here and you are now cutting it, then it opens up like so, and in between you could put another piece. If you just cut that piece that you want to add in with the same enzyme so that it has the same overhangs on the left and on the right, and if you then put that piece in, that new piece, and seal the ends with the enzyme ligase that you already know from the previous lecture, then you can create new pieces of DNA. So you can splice pieces of DNA together in the test tube. That's why restriction enzymes are a really, really powerful tool for recombinant DNA, for making synthetic pieces of DNA. Okay, so here's our box again, our gel electrophoresis box, and it's next to a circular piece of DNA. Circular pieces of DNA are often found in bacteria, and there they are called plasmids. Bacteria have their main DNA, the chromosome, but they also have these small plasmids, and they can hold a few genes, and they can easily be swapped between other bacteria. And that's why they are so powerful for bacteria. They usually have some sort of a gene on there somewhere that's important to them. For example, an antibiotic resistance gene that allows them to live. They only need it if they live in an area or an environment where they otherwise couldn't live, for example, in an antibiotic. But if they have the resistance gene, then they can live there. So why would they carry that on an extra chromosomal piece of DNA? Well, maybe normally, all their life and maybe all their offspring's life, it's very unlikely that there will be that kind of pressure from the environment to have the resistance. And so you might only get that resistance from a colleague, another bacterial cell, and then start to replicate it rather than replicating a bunch of stuff that you never need. So anyway, plasmids are circular pieces of DNA, and they originate from bacteria. In biotechnology, which is what this lecture is about, we use natural things that are already there, like plasmids, and we use these for holding pieces of DNA and letting the bacterium amplify those pieces of DNA. So a plasmid can be used to hold and amplify synthetic recombinant or any kind of natural gene that you cut, place somewhere in between two cut sites of a plasmid, a circle that you open up, stick in the piece that you want the bacterium to hold, close it up again with ligase, stick it back into the bacterium, and then let the bacterium keep amplifying it. It's much easier than doing it by polymerase chain reaction, for example. So why is that on here, and what does it have to do with gel electrophoresis? Well, these plasmids, when you cut them, they have restriction sites, right? They're made of DNA, so you just need the right enzyme in order to cut the DNA of these plasmids. And this is an example where each tick mark represents a cleavage site or a cut site for the enzyme EcoR1. So that's one of those enzymes that can be used. And so if you have a plasmid that has these cut sites here and here and here and here and here and there, those are the places where it can cut, then that means you will get pieces of DNA after cutting this entire plasmid up with that enzyme of the following sizes. This is 5.5 kb. This is 4 kb, so 4,000 base pairs. This is 7.5 kb. This is 5, 6, and 22 kb. And they fall into these pieces because this particular plasmid, as an example, just happens to have these EcoR1 marks here and here and where it says right here. Now, if you have this mixture and you want to just take out a piece, maybe you just want the largest piece and you want to splice that together with another piece somewhere, then you can take that whole mixture, run it on a gel, and you will then see the different sizes of these pieces that will run on the gel according to their length. And so that piece here is this small piece down here. And that large piece over here is this large piece. And now, if you wanted to, you could take a scalpel, cut out that piece of DNA from the gel, get rid of that agarose gel that's around it, and then stick it together using restriction enzymes and ligase. Ligase puts two pieces of DNA together if they fit, and make your own new piece of DNA. So that is then called a recombinant DNA because you recombine it. You are the one recombining it. 2 times ago in lecture, we talked about recombination in the cell. This is an example of recombining pieces of DNA to make synthetic new stretches of DNA. And then you can use the enzymes, you can use the plasmids for storage, you can then use gel electrophoresis for identifying the size of specific pieces that you want to use in your recombinant DNA analysis. Your book shows another technique that used to be really, really important and powerful, and it's called Southern blotting, and it's barely used anymore. That is because the DNA sequencing technology has become so powerful that this particular tool, this particular technique here, is not so important anymore. So I used to teach about this all the time. I used to do it in the lab myself, but now really hardly gets done anymore. It's good to know what it is, but sort of also know that the guy got a Nobel Prize for it. His name was Southern. So it was really important maybe 30 years when we first started to learn about identity of a gene but it's not really used that frequently anymore. So let me go through this relatively quickly. It's good to have heard about it. You probably will never do it. So what it does, that is important to know. Why do you do Southern blotting? You use it to determine the presence or absence of a specific gene. And the reason is because you are using a complementary piece of DNA to a piece that you're looking for in a preparation of DNA. And you're basically making those two pieces bind, and the one that you start with, it's called a probe because you're probing that unknown solution of DNA. That is labeled, that will bind if that piece is available, if it's there in the mixture, and then it will light up on a gel like so. So these two bands here will say, yes, in this preparation, on this lane of this particular DNA preparation, that particular gene that I was looking for is there in two copies, a larger one and a smaller one. Okay, so piece by piece, how does this work? You take DNA, let's say you extract DNA from a sample, you want to know, is gene X in this sample? You now add restriction enzymes to the sample. The DNA gets cut up, otherwise you only have the whole chromosome in there. That won't really tell you much, and it also is too large to really run in a gel. It will just stick in the very top in the well and never really leave the well. So you then run it on a gel, these pieces. You take that gel and you make this Southern stack. And so there's a tray on the bottom, there's some buffer in there. And then you put your gel in there, you put some filter paper in there, and you put a membrane on top. So after electrophoresis, the DNA fragments in the gel are denatured and then transferred, that's where the blotting name comes from, onto a filter paper and a membrane. The membrane, so they call the membrane the filter paper. So the liquid will take the DNA from the gel and it will keep moving upwards just because it's wicking, right? There's wicking paper on top. Usually there's a stack of just paper towels on top of this. And that wicking action will take the DNA out of the gel and deposit it on the filter. After blotting, the filter is removed. That filter is then stuck into something that looks like a Ziploc bag, and that's actually what people used to use about 30 years ago. And then onto that membrane, which is now a copy of the gel, of this DNA gel that was originally used. Now the membrane has the DNA. Now you're taking what's called labeled probes. So it's a piece of DNA that you make by PCR, for example, and you incorporate some radioactive label or fluorescent label. So that label is maybe nucleotides, let's say guanidine, that's radioactively labeled or fluorescently labeled. And then it will bind to the DNA that it will find on this membrane. And wherever it binds, it will light up. And so if it's radioactive, then you use X-ray film to expose the radioactive film, the radioactive filter to a piece of radioactive film. That blackens it and allows you to see it. Or if it's fluorescent, you can take a picture of it. Here's the bottom line of a Southern blot. So Southern blot is used to allow you to understand whether or not there is a presence or an absence of a gene and the number of the copies of that gene in a specific preparation. And you can see here that, yes, it is there in all of these 6 bands, in all of these 6 lanes, because you see bands everywhere. There's also something about the sizes and how you can use the sizes for for more information, but that goes way beyond what I want to tell you about today. So if you just know Southern blots were used to determine presence or absence before whole genome sequencing was done, then that's all you need to know. Speaking of genome sequencing, so I'm not going to talk about whole genome sequencing, but it will be great for you to learn how traditional sequencing was first developed, and that gets still used. So traditional sequencing of DNA is using small pieces of DNA, maybe a KB, maybe 1,000 base pairs at a time, that are sequenced. And that uses a process that's called Sanger sequencing. And Frederick Sanger, just like Southern, I forget his first name, both of them got Nobel Prizes for their discoveries because the techniques that they founded with their inventions, if you will, are so elemental for biotechnology that they were really huge when they were first developed. So Sanger sequencing is something I do want you to know how it works. And I'm going to show you a little animation at the end. I'll first walk you through it, then I'll show you the animation. And then I urge you to look at some other animations online. There are plenty of them on there. Some are quite long, some are pretty short. Watch a few and I think you'll start to understand how this process works. It's not so straightforward, but after you've seen it a few times, I think you will understand what the gist of it is, how it works. All right, let me try to explain. So if you have the principle is based on the fact that you are elongating a strand of DNA, basically like PCR, except you're only going in one direction. You're not doing the back and forth. You're not doing multiple cycles. So if you extend a piece of DNA, just like you would in DNA synthesis in the cell, you take a nucleotide, like this one, and you add it to a previous nucleotide, to the end of a DNA strand. Right? So this can have a growing strand, this can make a growing strand by putting this piece of DNA onto a previously existing dinucleotide. So here it says a normal deoxynucleotide has a hydroxyl group here on the 3' carbon, 1, 2, 3, 4, 5, so 3' carbon, allowing this end to be elongated. So what this piece is not showing you are the bases, right? The adenine, guanine, thymidine, cytidine bases. So it's just showing you the sugars and the phosphates. So sugar, phosphate, backbone. This hydroxyl group is required for you to elongate this chain and add on a new nucleotide. So if you are doing PCR, you have to add nucleotides into the solution because the enzyme will need to take a nucleotide and stick it onto the growing chain. If the cell is synthesizing DNA, it has to do the same thing. So that happens in the cell, and the enzyme will take a nucleotide that was made in the cell and stick it onto the growing DNA chain. In this technology of the Sanger sequencing process, you use not a normal nucleotide. So these normal nucleotides are called deoxynucleotide because they're deoxy here, no oxygen here on the 2nd carbon, but they have an oxygen on the 3rd carbon. Instead of using this deoxynucleotide, also abbreviated as DNTP, what you use in Sanger sequencing is a mixture of DNTPs and DDNTPs. So this one is a dideoxynucleotide because also on the 3rd, it's missing the oxygen. So on the 2nd, it's missing the oxygen. On the 3rd, it's also missing the oxygen. So it's a dideoxynucleotide. Now, why would you do that? If you have a dideoxynucleotide, you cannot elongate the chain anymore. Well, weren't we trying to elongate the chain? That's why we were using the deoxynucleotide. So if you use mostly the nucleotides that can elongate the chain, and every now and then basically you add a little bit of this dideoxy in there, and the chance, because you dilute it so much, is relatively small that the enzyme will pick this by accident or design and add it to the growing chain. Then only at the time when it grabs this will the chain be terminated. So this is called a chain terminator. The DNA chain is terminated. So Sanger sequencing uses dideoxys. It also uses normal deoxynucleotides, and the dideoxy cannot be used for elongation. That's the point. Now let me show you why that's the point and why that works well. So if you have a template strand, and here it shows you the sequence, but you wouldn't know the sequence if you want to sequence it, right? That's the point. You don't know the sequence, you want to know the sequence. But let me show you with this example first, with known strands, known pieces, what would happen. Here's your template. And here you have a growing strand of a piece of DNA that is now making a copy. So it's just like in the cell, or it's just like in PCR, where somewhere up there there's a primer, and this piece is growing and extending on nucleotides towards the end. And so at this end, another nucleotide will be added unless the enzyme picks a dideoxy. And as soon as it picks a dideoxy, it terminates that, and that's the end of the strand formation. Now, these dideoxys, not only are they terminating the process, they also are fluorescently labeled. And they're fluorescently labeled with colors that indicate whether that nucleotide is a C, a T, an A, or a G. So T's are maybe fluorescently labeled red, A's are fluorescently labeled blue, what is this, green, with a blue, with a purple, different kinds of colors, but for every same letter, same color. And these are only labeled for the dideoxy, so you will only ever label the end of a growing chain. So the sequence of the template is unknown. The elongation of the daughter strand stops whenever a dideoxynucleotide terminator is incorporated at the 3' end. In this case, each of the possible dideoxynucleotides is labeled with a different dideoxy. Okay, so then in the second step, now you have a whole mixture. So this is just to show you how that ends right at the end. But if you have a mix of all four DNTPs and DDNTPs, then you will create all sizes of all strands, because in most cases the enzyme will pick the DNTP and it will just simply elongate the strand. But every now and then, maybe 1 in 100 times or so, depending on how long your strand is that you want to elongate, the enzyme will grab the terminator. And so we will have all these pieces that are terminated of all different sizes and different lengths. And if you get the concentrations and the numbers just right, so how many DDNTPs to DNTPs you have, then you will have, for a piece of unknown DNA sequence, a number of pieces of DNA that all still have unknown nucleotides, with the exception of the last nucleotide. The last nucleotide will be known because you know that it was a DDNTP, and if it's green, then it has to be an A. So if we can determine that this piece is an A and has only 1 base pair long, and this piece ends in a G and it has 7 nucleotides, then we can say the first piece over here was an A and the 7th piece over here was a G, and likewise for everything in between. So how do you do that? Well, you use essentially gel electrophoresis. I say here that in the second step, you separate the mixture on a gel, but you don't do it on a slab gel like you do with a gel electrophoresis. You do that on a column gel. It's a very thin column. It's basically a plastic tube filled with gel. And so the DNA runs through there. Same principle, gets pushed through, separated by size. The smallest pieces will come out at the end, and there's a laser detector that can detect the fluorescence color of this piece. And so it runs through, the first small piece comes through, and the detector sees, there's green, and it tells the reader here to make a peak when it sees a lot of green color. And then the next piece comes out, and it's a little bit longer because it's now 2 base pairs long, and it ends, let's say, with a C. So then the fluorescent dye, the purple fluorescent dye, will register on the reader that it's a purple signal, and so then the machine will register that there's a purple peak. And so in the end, what the researcher gets is this, it's called an electropherogram. So you look at this and you say, okay, the smallest was a A, the next one was a C, then there was a T, then there was an A. So with that, you can deduce the sequence of the entire length of this unknown piece. And you can sort of put it together like that. Right? This is the A, this is the C, that's the T, that's the A, and so forth. But the longest piece indicating the end of this sequence being a T. And so then you can determine what the original sequence would have had to be on the other end. So once the daughter strand sequence is known, then the sequence of the template strand can be determined. That's called Sanger sequencing. That's super powerful, still gets used. We use it all the time at Puget Sound as well for research. This is sort of the way that DNA sequencing was first done. The first human sequence draft was actually done with this technique. Now, 20 years later, we have sequencing machines that go much, much faster, and maybe we'll have time to talk about those as well. But even though this is the classic method, this is an important method to know. And I have a little animation here for you that we can look at. And I would urge you to look for more animations, because that's not so straightforward how that works, right? So some of you will probably be scratching your heads. Let's watch this and see if that helps. And if not, go online and find some more that are maybe a little bit longer and show you this in 3D and sort of with moving pieces.
The first method of sequencing the genetic code was devised by Fred Sanger. To sequence the DNA, it must first be separated into 2 strands. The strand to be sequenced is copied using chemically altered bases. These altered bases cause the copying process to stop each time one particular letter is incorporated into the growing DNA chain.
Of course, those are the DNTPs that I talked about, right? All the question marks are regular DNTPs, so unlabeled nucleotides. But the labeled ones are the ones that are stopping the elongation.
This process is carried out for all 4 bases, and then the fragments are put together like a jigsaw to reveal the sequence of the original piece of DNA.
All right, so pretty cool. That is Sanger sequencing. Okay, let's talk a little bit about a process that's called cloning. Cloning is a term that's often used very differently in a DNA lab or biology lab, genetics lab, as opposed to out on the street or in the newspapers. So I say here that what cloning means for a geneticist is using restriction enzymes to splice pieces of DNA together and then to store them in bacterial plasmids and have the bacterial plasmids make tons of that copy. That's the process that's described here. I'll walk you through that in a second. The second definition of cloning is, what most people understand when they talk about cloning is making an exact copy of something, for example, an organism. So if I say, well, I have to clone myself, I have so much work, that means I have to make an exact copy of myself, and then there's 2 of me and we can do more work, or something like that, right? So cloning animals would mean make an exact copy of that animal or a plant with the exact DNA sequence. So that would be cloning. Now, those 2 terms mean something different, really. One is storing of DNA and making recombinant DNA, and the other one means making copies of something. But the origin is really the same, and that is because here in this last step, you make a ton of copies of the same thing in this process that a geneticist would also call cloning. So let me walk you through this, and then maybe it becomes clear. So earlier I said you can take restriction enzymes, and those restriction enzymes can cut the DNA at a particular sequence. And maybe you want to take a particular piece of DNA out and you want to store it somewhere, or you want to put it in a plasmid, also sometimes referred to as a vector if it's not in a bacterium anymore because you've dealt with it, you've done something with it. It started out being a plasmid from a bacterium that you took out of the bacterium, then you mucked around with it, and now you call it a vector. So if you cut that with a restriction enzyme and you cut that piece of DNA from a certain chromosome that you want to add into this plasmid, you cut that with the same restriction enzyme, then you see these little overhangs here. And you see that these overhangs here, if you use the same enzyme, will leave you with the same sequence. So this has an overhang of TTAA, On the bottom, of course, you would have the opposite strands, the complementary sequence AATT. And if you do the same over here and you cut this end and that end with the same enzyme, now you have an overhang of TTAA here and AATT on the bottom. And so, of course, this AATT would very nicely match up with this TTAA. And so that's why they're called sticky ends, because they are now sticky for each other, right? This and that can stick together and they will form bonds, hydrogen bonds, and then you can add some ligase and seal it all up. And the same is true as over here. So this piece of DNA can fit into this cut open plasmid or vector, and the resulting fragments are mixed together and joined by DNA ligase. The genomic and vector fragments have complementary ends. So these pieces are now in here, and now you have a recombined new piece of DNA, and it's called recombinant DNA. You use DNA ligase. Now you stick that into a bacterial cell, and I say you stick it in there, that's not very accurate. What you do, and actually you will do that in lab, is you will take bacterial cells and you heat them up just enough, you heat shock them is what it's called, just enough to make the membranes, the fluid membranes, remember that they're relatively fluid because of their the lipid structure of the lipid bilayers. So if you heat them up just enough, just like if you heat up butter and it becomes liquid or warmer, and then therefore more malleable, the membrane becomes a little leaky and it picks up plasmids from the outside. Doesn't know what it's picking up. It's hoping it's picking up something useful from the environment, and what it's picking up in the lab, of course, is the plasmid that you made with the recombinant piece of DNA. So now the bacterium will pick up this new piece of DNA. It will be inserted. It has its own DNA, of course, it's bacterial DNA, but now you add this plasmid DNA. That plasmid DNA with the recombinant DNA is now introduced into the bacterial cell, and then the bacterium will just do the amplification, because bacteria do that so very well. They just grow on plates or in liquid medium, and E. coli, for example, that gut bacterium, human gut bacterium I was mentioning earlier, that replicates itself every 20 minutes. So you come back overnight and you have thousands and thousands of new colonies or colonies of new cells that all have the same copy of this plasmid that you stuck into one individual cell made. And so that's where the copying comes into place here, right? Where we say that cloning means copying. That's really what's happening here in this process, except that a geneticist will call this whole process cloning, and sort of in the general language use, cloning just means copying sometimes of an organism. So now why would you do that? Well, you would do that because you might want to stick pieces of DNA together. So you could take another piece of DNA from a different chromosome, right? And you could cut it the same way and ligate it right here. So now you have a recombinant piece where you have two pieces that are from different organisms or different genes. Maybe the promoter, remember the promoter? The promoter is the switch that turns on a gene. So promoters are specific for their genes. They turn on the genes when they need them. Well, maybe you want to tinker with that. Maybe you want to see what happens if I put a switch, a promoter, in front of a gene that it normally doesn't regulate. Can I change the regulation of a gene by changing the switch that turns it on or off? You can do that by making a recombinant piece of DNA, hold that in a plasmid, stick it in bacterium, and actually see the bacterium is a living organism. So you can see maybe it does that job for you and shows what would happen if that particular gene is now under the control of a different promoter that you use. So those are ways how cloning is used for understanding the function of genes. Okay, one last thing, and that is the technology called CRISPR. CRISPR, you've probably maybe have heard in the newspapers about CRISPR. CRISPR is sort of the hottest thing on the market right now in biotechnology. It has been around for almost 10 years now. And what it is the harnessing of a bacterial mechanism that bacteria use for defending themselves. Similar to the restriction enzymes, right? Restriction enzymes are enzymes that bacteria use to defend themselves from bacterial viruses that infect them. Well, if they can cut the invading DNA, they can get rid of the invader. CRISPR is a similar process that bacteria developed over evolutionary time, and that researchers, biotechnologists, have now harnessed to do something slightly different, well, almost completely different with it. But it's based on this original system that was developed in bacteria. Okay, I'm not going to tell you about that bacterial system right now. That is very interesting on its own right, but I'm just going to tell you how that system is used for editing DNA. Why would you edit DNA? Well, there's a number of reasons why you could use CRISPR. Sort of the furthest in the future is, can we use this mechanism to edit genes that are bad, that contain a disease, that create a disease? Can we edit a disease gene to become a regular non-disease gene in an organism, maybe even in humans? There's all kinds of ethical questions about that. But right now, we just want to talk about the technology. Can we use this to edit the DNA of an organism? It doesn't have to be human right away. Maybe it's a mouse. Maybe it's a crop plant or a crop agriculturally important animal. All right. So right now, we're just doing this all in a cell. This cell could be grown into an organism. But right now, I'm just going to explain this for you, how this happens in a cell. So this cell has to be assembled. So you take a cell, you put some things in through transformation. You will learn how to do transformation in the lab later on. Let's assume this cell has what we need. Then what it has is the piece of target DNA. That's the DNA that we want to edit. That's the blue piece down here later on. Then we have a piece of DNA and we hold it in a plasmid. So this plasmid contains the editing template. We could just draw it as another stretch of long piece of DNA, the purple or pink one here. So that is what we want the bacterial cell in the end to use as a template to fix what we're going to break in this target DNA. And then we need the part from the CRISPR original machinery from bacteria. Specifically, we need an RNA that has a certain structure. This is called a hairpin structure. It's a single-stranded RNA, but here it's double-stranded, as you see, but it's only double-stranded with its own little curve that it's making. And an end, and that end is complementary to the target. So in bacteria, that would be targeted towards maybe a virus that it is immune against because it has this RNA sequence. But in technology, we make this ourselves. We make this RNA sequence, and we use that sequence strand down here, and we place that with the sequence of the target. This is our target. Maybe there's a gene that we want to edit. It has a certain sequence that makes you sick, and we want to get that out. So we now have the piece of that DNA that we want to target. We are, in addition to that RNA sequence, adding an enzyme that's called Cas9. And we have our cell here produce that Cas9 protein. We just put the DNA in here. We ask it to be transcribed and translated into Cas9 protein. And that Cas9 protein then is this brown thing here, right? These brown blobs. They take the RNA and they now go searching around in the cell until they find a piece of target DNA that is complementary to the search strand, to the RNA strand. So it binds, it unwinds the DNA there, and now the Cas9 enzyme, as it's binding the target, using the RNA as a target finder, it cuts that DNA. And it cuts it on both ends. So you see a cleavage. It has an exonuclease activity. That means it cuts the DNA. So now the DNA is cut. And now the template comes into place. Because whenever DNA is cut, and that can happen in normal cells due to UV irradiation, for example, or to chemicals in the environment, that the DNA breaks. So that happens relatively frequently, but we have repair enzymes that can fix that. And they will fix that. They now use an enzyme that's also an exonuclease. And that exonuclease takes this piece of DNA and just sort of cuts it nicely to the end, because there might be a little frazzled, there might not be the right overhangs, that could be simply stuck back together. So it just chomps it back a little bit, makes it blunt-ended, and then uses enzymes that are not listed here to put it back together. But if it does that, if it just puts it back together, then we create a mutation in this gene, because now we're taking out a chunk, right? That whole chunk is suddenly missing of, what, 10 base pairs or so. Well, that would mutate that gene for sure. So if we want to repair the gene, then we can ask the cell, why don't you use your DNA polymerase that uses a template to determine what to put in terms of a template, just like you do in DNA synthesis, right? You have the DNA strand, one is the template strand, and the other one gets added in new. So here we're providing a template strand. We're asking the enzyme in the cell to look at that and basically copy that into this piece that is resected, that has the gap. And as it's doing that, it puts in this light blue new sequence using that template strand. And if that template strand is exactly the sequence of the normal target minus that mutation that we want to get rid of, then the new piece is edited and the sequence is repaired. And so that results in an edited DNA with an altered sequence. Now, you could spin this forward in all kinds of different ways, right? You could edit a piece of DNA that isn't mutated. You just want to mutate it in a very specific way to alter its function just a little bit. You could do that using this technology. Maybe you want to get rid of a gene and you just want to mutate it so that it's no longer active. You could do that with this technique. You just have to omit this template DNA. We would just let this stand as is and then ask the repair mechanism to just fuse it together, and then you have the chromosome still intact, but the particular piece of DNA that was maybe normal here, you have mutated using that technology. So CRISPR is a very, very powerful technique for making mutations or repairing mutations or changing genes in any which way that you want to try to change the function slightly. So let me summarize what we discussed today here in our biotechnology lecture. I started with polymerase chain reaction. I told you that's used to amplify DNA. We talked about gel electrophoresis. We had talked about that before, but we said that that is used to separate DNA molecules by size and to make them visible. Restriction enzymes cut DNA. Plasmids hold DNA. Southern blots are used to look for presence or absence of specific DNA sequences. Sanger sequencing is used to sequence and deliver the sequence of an unknown piece of DNA. Cloning is the process of sticking pieces of DNA together and putting them into a plasmid using restriction enzymes and potentially amplifying that in a host, usually a bacterial host. And then CRISPR technology uses a natural bacterial defense mechanism to edit or mutate DNA in an organism. I know that was a lot, but it's really exciting stuff, you have to admit. So I would suggest for these technologies that are new to you, look at them online, get some animations and just watch a few animations, because the more different ways you see this happening in 3D in front of you, the easier it will be for you to understand. And then several of these things we will do in lab hands-on as well. All right, that's it for today. I will see you in class.